Hi, everyone. I'm John. We'll be starting shortly.
Hello. Just 1 second. Dieter, the slide, please. Okay, I think you can start. So, hello, everybody, and welcome to our webinar, Pain Points and Spectrophotometric Workflows. My name is Merlin, and I will moderate today's webinar. I'm seeing we have a lot of people who joined already, but let's give the others who signed up maybe 1 or 2 more minutes. Maybe in the meantime, where's everybody from? Maybe you can write the country where you're from in the chat. Okay, we have one from Turkey. Don't be shy, people. Germany, Bangkok,
Thailand, yes.
Very international.
Oh,
South
sorry.
Africa. Indonesia. Many people from Indonesia. I've seen this in the signups already. Nice. South Korea. Okay, I think we can start the session. So, like I said, my name is Merlin. I'm from Tip Biosystems. A short introduction. I oversee operations and manage the marketing and sales here at Tip Biosystems. Then we also have John and Dieter here in the call. Maybe John, if you can give a short introduction about yourself, please.
Yes, hi. Hello, everybody. I'm John Piankowski. John is good enough. Recently joined Tip Biosystems as general manager. We're excited because this is the first of our next series of webinars. We're on the move as a company. We have a lot of new things that we're developing, and we hope that you get a lot out of the session today. And please remember, if you didn't submit questions already, please feel free to put those in the chat, and we'll address those at the end. Okay.
Okay, Dieter.
Okay. Yeah, thank you, Merlin. So, yeah, good evening, everybody. My name is Dieter. I'm originally a German engineer, but I'm also a professor in analytical chemistry and biotechnology. I taught in Hong Kong at the Hong Kong University of Science and Technology, and in Singapore at the National University of Singapore. And now I am teaching at the Asian Institute of Technology here in Asia. I started Tip Biosystem with my team in Singapore in 2015, where we developed, a very exciting new spectrophotometer, which we will explain later in this webinar. Thank you.
Okay, so, thank you, Dieter. Yeah, maybe a few words first. So in the webinar registration, we asked everybody to submit their pain points, and the results were actually quite interesting. Several pain points were mentioned multiple times. And in this webinar, we are trying to address those pain points that, that seem to be the biggest problems. If we don't address the pain point you submitted, don't think that we ignore it, but there were so many submissions that we simply can't address each pain point individually. But after this 1 hour session, this 1 hour webinar session, we will have a Q&A at the end where you can again address, write your pain point in the chat, and then we can try to answer it, or also any questions you might have. If you have questions during the webinar, feel free to write it in the chat. If we can, we will answer it during the session or then later in the Q&A. One ground rule, please remain muted and keep your camera turned off. Thank you, everybody. Okay, Dieter.
Okay, thank you, Marian. Then let's start with the webinar and pain points in spectrophotometric workflows. Where we want to address those that you have submitted, and we also want to introduce you, a new type of spectrophotometer and workflows that you may consider in your company or in your studies. So let's go first. Let me introduce a few terms, and there's sometimes quite some confusion. What exactly means, what is a spectrophotometer, what exactly is a photometer, and of course the technique, we call it spectrophotometry and photometry. So let me start here very simple. So a spectrophotometer is a spectrometer that is for the optical region of the electromagnetic spectrum. So sometimes I hear people, they say they mean a photometer has a single wavelength and the spectrometer is an instrument that records a spectrum. This type of thinking is very often used, but it's actually not really correct. So a photometer actually is an instrument that measures light intensity. Yeah. For example, the entire light intensity of the sun or the light intensity of a light bulb. Actually, a photometer does not really have a light source. It measures the surrounding light that is there. Yeah. And here on the slide on the top right, you see a picture of such a photometer. Sometimes people also call it lux meter because the unit of the light, intensity is a lux. So those instruments, they are used in meteorology, in astronomy, and if you are a photographer, you will also use them to measure the light intensity of the scenery you are going to photograph. So now, if we would add a light source to such a photometer, and if this light is of one particular wavelength, what we call monochromatic light, then it actually becomes a spectrophotometer. So the photometer will become just the light sensor. And and here's a picture on this slide, on the lower right side. This is a typical small, a spectrophotometer, and I give you an example of an instrument we often use in the biolab. This instrument measures OD600. So it only has one wavelength, actually 600 nanometer, but it's actually still a spectrophotometer because it only measures, this small part of the spectrum. So now let's go to, what is actually a spectrophotometer. So a spectrophotometer is registering the light intensity over a certain, area of the spectrum. But it doesn't actually matter. It's maybe one wavelength, three wavelengths, or, it records the entire spectrum. Let's say in the UV region or the visible light. So on the top right here, you have a picture of a typical spectrophotometer, and we come back to this later, how they exactly work. So usually if we say, spectrophotometer, we imply that it's an instrument, mainly a benchtop instrument that contains its light source, and it contains something that's a monochromator and so on. Sometimes there's also this term spectrograph. So what is a spectrograph? Actually, a spectrograph is an instrument that records the light intensity over a certain area of the spectrum. And those type of spectrographs, they are used in astronomy or study the sun, what type of rays the sun is emitting. Okay, now let's go a little bit deeper. How is the spectrometer designed? What are the components? So here we see a typical setup, of a spectrophotometer. So as mentioned, it has a light source, but most spectrophotometers, their light source it's not monochromatic light. It's white light, and white light contains all the different colors and all the different wavelengths. So typically this light source in a spectrophotometer, we call it a tungsten lamp or something like this. And then we mentioned, we want to study the light at a particular wavelength. So this means we have to split the white light of the spectrophotometer into one particular wavelength. And this is usually done with a device in the spectrophotometer that we call a monochromator. So the monochromator creates monochromatic light, so light of one particular wavelength. And that's shown here in the picture. I hope you can see my cursor. And only this monochromatic light, in this case, this yellow light, will then enter the sample. So here we have the sample, and then the light is transmitting through the sample and reaches the detector. And the detector simply measures the light intensity that arrives at the detector. So now we have a sample here, and typically this is liquid samples. And so in this case, now the sample could contain, let's say, water, but water does not absorb white light. So this would mean all the light would actually transmit through the sample and is reaching the detector. But as soon we have any substance, or it could be also particles that scatter the light, or bacteria cells, if we have some in the sample, some of the initial light intensity, which here I0, that's our initial light intensity, will be actually absorbed or scattered, and therefore will not come out here and will not reach the detector. So in this case, the initial light intensity is, we can call it 100%, and only a certain percentage of this is reaching actually the detector. So with this, we mentioned already, typically with a spectrophotometer, we measure liquid samples. And here on this slide on the right top, you see a typical spectrophotometer wire linked to a computer or notebook to control the instrument. To get your sample inside, you have to fill your sample into a cuvette. Those cuvettes are typically made from plastic or from glass. If you measure in the UV region, you need a quartz glass. And then you need to fill your sample into the cuvette, place the cuvette in the spectrophotometer, and take your measurement. Actually, it's not limited to a liquid. Actually, spectrophotometers can also measure gases. And here you see another cell. It's basically a cuvette. It's a cell where you can pump in a gas, and you can measure the absorbance of the gas. But typically gases do not absorb that much. This is why we have here in this cell a very long optical path. Maybe that's 10 centimeters. But our optical path here in our cuvette, the standard length is 1 centimeter. Okay, so now what do we actually measure? There are these terms that flying around, optical density, absorbance, transmissions. What does this mean? So you remember, this is a part we saw before in the setup of the spectrophotometer. Here's our initial light intensity coming in, and here's our sample. And if the sample has no optical density, if there's nothing in the sample that absorbs the light, then all the light coming in also pass through and come out. But what optical density means is there is a substance in our sample that absorbs one particular wavelength or different particular wavelengths or a band of our sample. This means less light is coming out than goes in. So now, and that is what we at the end measure. We want to measure how much light is absorbed, but we can also say we want to measure how much light is going through. and that's transmitted. So if all the light goes through, then 100% of the light is transmitted, and that we call usually T. Here in this little table, you see the percentage of T. If T is 100%, our absorbance is actually 0. And below here, you can also see, the formula A, that's our absorbance. Absorbance and optical density, it's basically the same. You can use both of these terms. Absorbance, the abbreviation is the capital A, and optical density, we call it a capital OD. So now the interesting thing that you need to realize in spectrophotometry is that the absorbance is actually a logarithmic scale. So you see this here in the formula. You see this log 10, and it becomes more clear if we put our attention to this table here. So at the absorbance of 100, so no light is absorbed. The transmission is 100%. No light is absorbed. Our absorbance, or A, is actually 0. So if 10% of the light goes through, means actually 90% of the light was absorbed by our sample, then our absorbance is a 1. And you see it's logarithmic. It goes 100, 10, 1. So if 1% of the light goes through, then our absorbance is actually 2. And now you can also understand why most instruments, they can only measure absorbance up to 2 point something or so, because you can now realize that an absorbance of 3 means there's only 0.1% actually of the light that goes through. So now you can realize that your light detector must be very good and must be very sensitive. Okay, so absorbance is a logarithmic scale, and transmission is just the percentage of light that can pass through your sample. Okay, so how does look spectrophotometers today? So here we also want to address some pain points that were submitted. Typical spectrophotometers today are benchtop instruments, like in this picture. You see them here on the top, and for a very, very, very long time, for decades and decades, that was the standard. And there were, in my opinion, that's my personal opinion, 2 big advancements in spectrophotometry. One very big advancement was the introduction of an instrument called a nanodrop, where the sample volume was dramatically decreased. In a typical spectrophotometer, the cuvette, which I showed you before in the slide, it's usually 1 milliliter sample that's required. And with a nanodrop, that amount of sample was reduced to just about 2 or so microliter. So, and this is a picture here in the center top of such an instrument, which is still a benchtop instrument, and it's still wired to a notebook computer. So that's one of the pain points, that the benchtop instruments need large sample volumes, and they also need certain calibrations, calibrations regarding the wavelength accuracy. Do you measure really the correct wavelengths? Because such instruments, they have moving parts and so on, and the wavelengths can maybe change. So this problem with the wavelength change and so on can be addressed by, an instrument using filters, or an instrument similar that we built, which uses an LED, a light-emitting diode, because they do not really change the spectrum. Another pain point of contemporary instruments is the warm-up time, and if you remember the setup of the instruments, which I just showed you, there's a light source. And as soon as you switch on the instrument, that light source is also switched on. But the intensity of such light source, they are a function of the temperature. So this means the intensity is actually changing over time. And if you remember the formula, that actually the intensity that goes in is important in calculating the absorbance. Then if this intensity changes, that will cause an error. So time can be one light source of choice here is, of course. So another pain point, that lab station, insert your sample into the lamp, connect your instrument to the to your PC and control it through the PC and perform your measurements. So there are several types of pain points here in this workflow. First of all, it takes quite a very long time. The workflow is stationary, so you need to go to the lab. And for certain samples, your sample transport can cause quite a big error. Let's consider you have a sample containing algae or something like this, and you want to measure the chlorophyll or the algae density. At the time you transport the sample to your lab, the algae may still continue to grow. And actually, your measurement in the lab might be very accurate, but actually your sample has already changed. And that's what we call a systematic error. Okay, so this type of problem was actually addressed by Tip Biosystems. And we have developed a handheld spectrophotometer, which you can see here in these pictures. And the handheld spectrophotometer enables you to measure on site without actually a transport of the sample. And there's also no sample transfer into another container, such into the cuvette, yeah, because the device itself contains its own cuvette. So here are some examples of applications. On the left, this is a cell culture, hood. Here, this green box, this is a special hood to work with nucleic acids, RNA and DNA. And then we have a scenario here in a brewery. And we have here a measurement outdoor, directly in a pond or something like this. And at this point, I should mention that if you would do this measurement at a pond or on a river bank, you would automatically also record your GPS position. And you can overlay your GPS and you can overlay the measurement in Google Maps. And so you cannot mix up the sample or the sample location where you have obtained these results. So that's why we call this, analyze anywhere, anytime. Okay, let's go a little bit deeper into this technology. We call this instrument a Photopette. So it's a fusion word from photometer and a pipette. And it's a system made up of 3 components: the instrument itself, the Photopette device here on top, and the instrument is linked through Bluetooth with a smart device. Could be either a tablet or a phone. However, there's no phone line necessary, and there's also no internet necessary to operate because it goes through the Bluetooth link. And the third component in this system is a cuvette tip. We call it cuvette tip. This is a disposable tip which is attached here to the end of the Photopette device. And this tip is replacing the classical cuvette that you use in the benchtop instruments. And your sample will only come in contact with this tip actually, here's a window, here's a tiny window, and there's no contact of your sample with actually the instrument itself. So you see, this is it's a closed system that works seamlessly together. And because this instrument has LED light source, and there's actually no moving part or anything in it, you know, so it does not really require continuous calibrations. It's factory calibrated and very stable. So yeah, we talked about this already. So this is another pain point, the instrument calibration, which we actually which we do not require with these instruments, but it's required with standard benchtop instruments. So let's go a little bit deeper. How this Photopette device works. It's actually, how does the light go to your sample? You know, the light is generated by LEDs, and those LEDs, they are linked to fiber optics. And these glass fibers, they actually transmit the light to the end, you know, of this stem made from stainless steel. And if you would have a closer look to this stem, you would see there are actually 3 tiny fibers, and there's 1 bigger fiber. And the 3 tiny fibers, they transmit light from 3 different LEDs. So they could have 3 different wavelengths. And the big fiber here, this is picking up the light that is transmitted through the sample and then directs the light to the detector, which is in this instrument. So you see, the instrument contains everything that that I showed you before in the setup. It does not need the monochromator because the LED itself is already the monochromator, and the cuvette tip helps that the light is passing through the sample. Because if we insert the cuvette tip in the liquid sample, here this blue thing, let's say here this is a reaction tube, then the liquid will actually flow into a cavity of the cuvette tip, and the light can pass through this. I think we have a little animation here. Yeah. So this is the light. So the light comes out of the fiber, passes through the liquid, and at the end of the tip here, there's actually a mirror coated from aluminium, and this mirror is reflecting the light back to the same sample to reach the big fiber and then to reach the detector. Yeah. So this is very different to a standard spectrophotometers where you actually have to fill your sample into a cuvette, but in this case, the cuvette goes into the sample. And this is why we do not require a sample transfer. So let's talk a little bit about general stuff here. Again, you know, that was one of the pain points submitted to us, you know, how to maintain consistency and accuracy. You know, one important thing here is to consider errors. You know, what type of errors actually exist in spectrophotometry. You know, like in any other method, you have 2 types of errors. You have random errors and you have systematic errors. So random errors, they arise from certain uncontrollable things in the environment that you have and so on. They are a little bit difficult to reduce. They always happen. They're usually not that big. One way to get rid of them is to do repeated measurements of the same sample and so on, and taking an average of those measurements. So more serious are systematic errors. So systematic errors, it means there's actually a systematically wrong approach in your analytical method. So for example, you have chosen the wrong wavelengths. So you may have an assay with the reagent, and if you have chosen the wrong wavelengths, then you would do a systematic error. Another systematic error is you have just chosen the wrong method, and the wrong method does not correlate with the concentration of your analyte that you are measuring. More systematic errors can arise, from pipetting. You use the wrong pipetting volume or something like this, and there are actually many different sources of systematic errors. So you have to think very clear if you choose the right method, and if there are something there that can interfere with your sample, and so on. But the good news is that systematic errors can be eliminated. Sometimes, as I mentioned with your pipette or so, you can recalibrate the pipette to remove this error, or you can recalibrate the spectrophotometer for the right wavelengths, and so on. Another interesting thing to consider if you perform spectrophotometric measurements and in terms of errors is, at which absorbance you actually have the lowest error. Have you ever thought about this? So actually, if your absorbance is very tiny, let's say 0.002 or something like this, then your error is actually quite high. And then if your absorbance increases, your error actually also decreases. And so the absorbance with the lowest error, it's about in the region of absorbance 0.5, something like that. And definitely until absorbance of 1 or so, your error is still very small. But if your absorbance become very high, 2, 3, or 4, then also your error increases. And I think now you can understand why it's like this, because I showed you before the logarithmic relationship, of the absorbance. And every absorbance step from 1 to 2 means there is only 10 times less of the light going through. This is why at a very high absorbance, the error is relatively high. However, with the technology we developed with the Photopette instrument, we have a very high dynamic range. We call this dynamic range, the absorbance you are able to measure. Our dynamic range is up to 3, and with a relatively low error. And how we can achieve this? We achieve this by using not only one light intensity, but many different light intensities, and actually also very high light intensities. And then we have developed an algorithm that looks at all this data and calculates the absorbance. And through this algorithm, we achieve a very small error, even if the absorbance is very high. Okay, so this is one of the main, game changers that we introduced with the introduction of the, Photopette handheld spectrophotometer. In my opinion, I mentioned to you before, we had benchtop instruments for decades and decades, and there were 2 big advancements. So the nanodrop is one big advancement in terms of very small sample volume, but I also see the introduction of the handheld Photopette as quite some advancement in spectrophotometry, because it brings the instrument really to the sample and enables you to perform high quality measurements in a very simple way and workflow directly at your sample site, which could be in the hood, or it could be in the environment, or things like this. Yeah. So let me quickly tell you a little story here. I think it's on this one. So that's my time, Dieter's PhD student time at Hong Kong UST. How we came up with the idea of this Photopette system. Actually, as I was a PhD student in Hong Kong, I did a lot of protein separations using chromatography. And in chromatography, you basically separate proteins by their size or by their charge. And then you have many fractions, we call it fractions of a liquid, and they are fractionated in an instrument like this. You see here this glass test tubes, and there are about 100 of them. And after your chromatographic experiment, you have close to 100 tubes, you know, where your separated proteins are. But so you cannot see actually proteins. They do not absorb light usually in the visible spectrum that we humans can see. And you cannot know where is your protein actually. And at that time, I had to take every single of these tubes, you know, transfer the liquid into a cuvette, place the cuvette in the instrument, close it, measure, and so on and so forth. At that time, I thought, hey, how nice would it be if I had some instrument which I can just dip in, measure, dip in, measure, dip in, measure, and directly find my protein. And you know, finally, after so many years after, we actually realized this type of dream which I had at that time with the handheld, you know, Photopette system. And here you see how the instrument is used to measure directly in such a tube, in such a test tube, without, you know, transferring the sample. Yeah, so that's the story behind, you know, the creation of the Photopette device. So today we have developed this into a family of instruments, and we call them Photopette Bio, Photopette Cell, OD600, Tubility, and they cover different applications. The Photopette Bio is mainly to measure nucleic acid DNA concentrations, and DNA absorbs at 260 nanometer. Photopette Bio also for protein measurements absorption at 280 nanometer, and for certain, you know, enzymatic assays at 340 nanometer. And 340 nanometer has also another function. It's used as a background correction for very accurate measurements of nucleic acids and protein. So then here in this list, you see some other instruments, Photopette Cell, you know, basically for the cell lab, for optical density, and Photopette OD600, the classical 600 nanometer measurement to measure bacterial density, yeast, and so on, and tubility. But we are also quite proud as a company to build you actually any instrument of your choice with any 3 wavelengths between 260 and 1050 nanometer. And we have some very interesting customers applications here where we built specific instruments for them for algae farming and for the galvanic industry, nickel plating. We can build instrument that measures nickel or copper in such a galvanic bath, and so on. And I believe that's quite unique because, you know, usually you cannot really ask a company to build you one instrument. They will ask you, hey, how many you need, 10, 20, 50, 100. But we can actually build a single one for you. And we are also very interested in to explore new applications. If you have something in mind, please contact us. So also on this slide here, you see a list of the key features, you know, of the Photopette product family. You know, there are actually fixed wavelength spectrophotometers using LED, no warm-up time, and all these problems. The main advantage you have seen, there's no sample transfer, and because we are using this cuvette tip, if you're doing, you know, microbiology stuff or molecular biology research, if you change the cuvette tip, so there's no cross-contamination of your samples. We have a high absorbance range, high dynamic range. Measurements are very fast. They just take seconds. And one more interesting thing you may notice that, you know, we can actually measure in ambient light. We do not need to, like a spectrophotometer, close it for the measurement because we have an ambient light correction function inbuilt in our algorithms in the instrument. It's battery powered, lithium-ion battery, It can last for many, many measurements, for more than 20,000, and it's compatible to iOS and Android, devices, tablets, and phones. And with every measurement you take, you also save in your same data file. You save your date and the time and the GPS position, and you can even take a picture with this phone or tablet, and that picture is linked directly to your result. So then what you can do with the data, you can export the data as a file, you can send it to your email, or you can upload it to a cloud server. And that's also very, very fast. So that enables you to share data in almost real time, with your team. Okay, let me give you some examples, of measurements and applications that you could do. Here's one example, measuring mammalian cell density at 600 nanometer. And here, this is a cell line called HeLa cells. They are very popular, used in research. And we have actually calibrated our device, Photopette cell, with many different cell lines. So you do not need to do your own calibration. There are certain cell lines, there are inbuilt calibrations in this instrument. There are also inbuilt calibration for E. coli and yeast and spirulina, the superfood algae spirulina, which in reality is not an algae, but is a cyanobacteria. Okay, another a pain point we had received was complex software, often benchtop instruments, so they are connected to a Windows-based a system, and the software is rather complex to use, has many functions, and requires quite a little bit of training. So this goes all away with our Photopette app, which you can download from the Play Store or so for free. But without the instrument, there's not that much you can actually do with it. But here on this slide, you see some screenshots. So our software is quite intuitive. There's not really much training. There's actually no training required. This is your landing screen, and where you connect through Bluetooth through a Photopette device, and then you can choose what we call measurement type. So if you click on the measurement type, you come to this next picture here, to this next screenshot. You can choose the wavelengths, or you can choose an application. So, because at the moment the app is connecting to the device, actually the device is sending its ID to the app. And then the app knows, this is a Photopette cell device, and this Photopette cell device has these type of wavelengths inbuilt, 340, 570, and 600, and then automatically list the supported applications here for you, and you can just click one of them. Here, for example, which I mentioned before, mammalian cell density, and then you can start your measurement. Of course, like any instrument, like any other spectrophotometer, first is you have to do an auto zero in either your buffer or water, and after this you can directly perform your next measurement. And this is all very fast. It's a very fast process here, because you do not have the sample transfer, and everything goes in seconds, and your results will then displayed immediately on the screen. And here's one example. In this case, the measurement type E. coli was chosen, and you can see the absorbance. You can also see the transmission, and you can see how many colony-forming units in this case, how many E. coli cells are present in your sample per milliliter. Yeah. So In this example here, if let's say you choose E. coli as measurement type, you can also do continuous measurements. Actually, we have a function that you can program. For example, you can program, I want to do 600 measurements every 2 minutes or something like that. And then the device will do this automatically for you or overnight or something. And this is very handy because we have 2 different ways how you can display your data. The table view here, but we also have the graphical view. If you in the app click on this little graph here, on this little icon, then it will switch to graphical view. And here in this case, you directly see your growth curve, you know, of the E. coli cells. So this is very, very easy and very simple to do. I must say, basically now we have achieved that everybody who can use an app can basically perform, you know, high quality spectrophotometric measurements, either in the lab or in the field or in the factory. Okay, so let's sum up a little bit, you know, on the value propositions here, you know, of the Photopette device family. You know, one of the biggest advantages, of course, is the ultra portable. It's ultra portable handheld device. It's very ergonomically formed. It's just 180 grams, and it's very convenient. It's wireless connected through Bluetooth. It doesn't need sample transfer. Allow real-time data recording, and measurements are very fast. It's just in seconds. It has a good quality of accuracy, repeatability. It has a high dynamic range. It's quite a robust instrument. As I mentioned, there's no moving parts. There's nothing in there. It's splash proof, and it's suitable to use it in the field outside. It's quite affordable. It's only a fraction of the cost of a conventional, you know, benchtop UV vis spectrophotometer, and it's maintenance free. No more cost here. It's also a smart device. It's basically a IoT ready, you know, device, and you can share your data in quasi real time, you know, with a cloud. It's innovative, you know, it opens some new applications. And as I mentioned before, we are very interested in some unexplored applications you may specifically have, you know, in your company or in your research, you know, then please contact us. Okay, so let me come now to last part of this presentation, some new products we are very excited to launch very soon. We want to add one more family member to the Photopette family. We call it Photopette Aqua, and the Photopette Aqua will enable you to measure a nitrate, nitrate, a phosphate, ammonium, and chlorine by using actually reagents from Merck, the company Merck, Millipore, and they have a reagent line called SpectroQuant, and SpectroQuant is a trademark of Merck. And we are using these reagents together with these instruments to measure these parameters, which are the most important parameters, you know, for water, for drinking water, and also some type of other industrial and process water. And chlorine, of course, for disinfection, where you do not want to have traces of chlorine in a certain of your water for certain processes. So we are intending to launch this at the end of this year. And yeah, please contact us if this is of any interest for you. We are doing now all the measurements, all the calibrations. And again, this instrument will have inbuilt calibrations for those analytes that you see here in the list. And this is a start. We may add, you know, more analytes later to this list. Okay. Also, I'm very excited about another development that we have. And again, we intend to launch end of this year. And this is a new product family, which we call a Spectropot. And the first member of this family will be the Spectropot UV. And this is a microvolume spectrophotometer, which only requires about 2.5 microliter of a sample. And it's supporting the wavelengths 260, 280, and 340. So again, to measure, nucleic acid, DNA, RNA, proteins, and 340 as a background correction. This instrument is ultra compact. It's quite small. It's lithium-ion battery powered, so it does not need a cable or anything. You can place this also in a hood or in very small lab spaces. You can work with this. And similar to the Photopette, this instrument is Bluetooth connected through our app. And so this app is also quite similar to the Photopette app. It's very easy and intuitive to use and allows you also very fast data sharing. So with this, let's maybe recall a few of our key takeaways for this webinar. What we have touched, we touched on what is really the differences between a spectrophotometer and a photometer, and so you have learned basically a spectrophotometer measures light intensity or light changes of your sample at one particular wavelength and monochromatic light. The photometer technically is an instrument to measure all your surrounding light, the sun intensity. It's used, maybe to calculate for a solar farm, how much power it should make. Then you would need a photometer. And we also have touched on how spectrophotometers work. So you remember they have different components. They have a light source and making monochromatic light, sending this through the sample, and then they measure the amount of light that was transmitted by a detector. And using this calculation, they will give you an absorbance value and a transmission value. So we also touched on errors, so systematic errors and random errors, which could be there. And so workflows, we have the traditional workflow with the benchtop instruments where you fill in your sample in a cuvette. And we have the new workflow with the Photopette, where actually the instrument goes to the sample and you are enabled to measure directly on site without sample transfer into cuvette, and also without sample transfer to your lab. Okay, so with this, you may be interested here. We have developed many more applications. I showed you some of them. And they're all available on our website at tipbiosystems.com, and and my team can maybe put also the link of the website in the chat for everybody to make a copy, or here's the QR code. Okay, so with this, I want to thank you all to be patient and to listen to our webinar that address some very general pain points in spectrophotometry, and also hopefully gave you some solutions of those pain points using our ultra portable handheld Photopette devices.
Thank you very much, Dieter. Thank you very much for this nice presentation. I hope everybody learned something, maybe something new and interesting. So actually our official, the webinar session itself is now over, but we are going to have a Q&A where everybody is welcome to join. If you have any questions, you can write them in the chat, but now you're also welcome to turn on your voice, your camera, and ask the questions directly. Okay.
And while we're waiting for questions, I can maybe remind everyone that we will be putting the recording of this on the Tip Biosystems website so that you can play it back again later on, or show it to anybody that didn't manage to see the original webinar.
Mm-hmm. Okay, maybe should I start to answer some question, because I just see some in the chat here. I could not follow the chat all the time when I was presented, actually, but now I see the last question here. Maybe I can start with this one. And the question is, I want to measure the sulfate in the water. Is the kit necessary for this? Yes. You know, this is interesting. This links directly to our device, which we are going to launch, the Photopette Aqua. And also sulfate and sulfate, there are some very interesting parameters, and you actually need a kit for this. You know, those ions, you know, sulfate and sulfate, they do not really absorb light much. So this means we cannot directly measure them. We actually need a kit that contains a reagent, and this reagent in some way will react with your analyte and then will form either a colored complex or something like this, which we can measure. Yeah. So yes, you need a reagent. You may also consider, you know, you can have a first look at the SpectroQuant reagents from Merck Millipore. They have kits for both of those analytes. I can maybe continue. Here's another question about the sample path length. The question is there concern of sample path length where actually 1 centimeter is used in classical spectro? What is it in this instrument? Yeah, that's a very good question. So all the results that we show in the Photopette app, they are equivalent to the 1 centimeter path length, which is standard in the industry. You know, you have the same thing with a nanodrop or so, you know, also the path length is tiny, and it's not really 1 centimeter, but the instruments calculates, you know, what you display to the standard path length of a 10 millimeter, 1 centimeter. And that is the case also with a Photopette system. And it will be also the case with the new instrument we are going to launch with the Spectrophotometric UV. So the real physical path length, you know, is actually 6 millimeter. And in the Photopette system, the light is passing through the same sample twice. First it goes in, it's reflected by the little mirror, and then goes back. Yeah. Okay, let me continue here. Maybe I just go as long as I see a sample. So Errol is asking, are there risks involved in inserting the Photopette to the sample? No. I mean, of course, here also you have to be careful with the errors. If your sample, maybe you work in microbiology and you do not want to contaminate your sample, then I would propose you are using our sterile tips. So we have 2 types of tips, our standard tips and our sterile tips. Our sterile tips are sterilized by an industrial process using ethylene oxide. And if you would use this tip, so there can be no contamination of your sample. Actually, the tips are made from a very high quality, you know, polymer, and they do not change or do not do anything with your sample actually. Okay. Okay, we have one more question here. What is the market price for the Spectrophotometric UV? So actually, we do not have yet fixed actually the market price, but we will do this soon when we launch the instrument. And so please, if you send your email to a mailing, you know, as soon we have some market price and we will have a little launch flyer, we will inform you about this.
But
it will be priced attractively. So we can say that, John, correct?
Yes, it should be very competitive against the nano drop. That's all we'll say now.
Okay.
I've just posted our email address in the chat. If you want to stay up to date and get product updates, you can email us, and then we will keep you updated.
Okay. Okay. So, John, do we have some more questions during my presentation? Because I was not able to monitor this all.
Sure. No, the only other question that's in the chat is about blood analysis in the instrument.
Yes. So, blood, of course, is a very optical dense sample. The absorbance of blood, if you don't dilute it, is super, super high. So only with a light path, which is very, very tiny, you can measure something. But then what exactly you want to measure? So is it blood, or is it a serum or plasma? Of course you could dissolve the blood to a dilution where you can actually measure it. Between 0 and 3 OD, or you could have plasma or serum and also dilute it and measure at 280 nanometer, for example, the protein concentration or things like this. But almost, I think, no instrument really can measure in undiluted blood directly. And here the question is also what type of information you want to obtain from the blood measurement. Yeah. So I think it's a bit hard to go deeper in this. If you have some specific application in mind here, then please send us an email, and we will look into this. Yeah.
Dieter, maybe I can ask. There was some interruption of your voice when you were on slide number 8. And if it's not too much trouble, maybe you could go back to that slide, because there was some very important information there that people were really eager to hear. And then there was some interruption of your voice.
Okay, give me a moment. I go first to the slide number 8. So the slide number 8, this was about spectrophotometers, contemporary spectrophotometers. Let me share. John, you mean this slide?
Yes, correct.
Yes. Okay, so on this slide, I was talking about the state of the art, you can say, today in spectrophotometry. For many, many decades, we actually worked with, benchtop spectrophotometers of the type here on the top left, a picture in the slide. Those instruments are very bulky, big. They have a cable to a computer to control them. And, I also mentioned here that, in my opinion, there were 2 big advancements in spectrophotometry. Of course, there were many little improvements, many improvements. But I don't mean improvements here. I mean really game changers. And one was the introduction of the microdrop instrument, which you see here in the center. And the game changer here was very small volume, about just microliters. And another game changer is, in my opinion, really the Photopette family of products. And here's a game changer is a workflow. So you can measure directly at the site, at the point of sample, and with a high quality, and share your results. And so also on the slide here, what we can see is quite a tedious workflow in the classical benchtop spectrophotometer setup, where you collect samples, transport samples, fill them in the cuvette, place the cuvette in the instrument, and then connect your computer with the instrument, start the software, choose the right function in the software, and take your measurements. So it's quite is quite time consuming and tedious. And during your sample transport, there might be some error here, which is, we touched about error, which is, you know, a systematic error. In case you would have, let's say, algae in your sample, then they could still grow. And during the transport, actually your sample is changing. In the lab, your measurement is very accurate, but unfortunately, your sample has already changed. Yeah. So this is one of the potential error pain points here that comes with this workflow. Yeah.
Mm-hmm. Okay, thank you. And there's one more question from Thailand for BGC issue, basically using air for BGC. What if there's a blank solution for BGC? How does that work?
So what is BGC here meaning? Maybe can we ask? I can write back maybe.
Okay, let's see.
Background. There's already an answer in the chat. Background correction.
A background correction. So this could so the BGC here, it could relate to what we explained with the Photopette Bio, 260 nanometer, 280 nanometer, and 340 nanometer. So those wavelengths are used, and the 340 wavelengths is used for this, you know, background correction, because proteins and also DNA, they do not absorb at 340 nanometer. So what the background correction basically does is, it measures, let's say we measure DNA. It measures at 260, but it also measures at 340. And we know DNA is not absorbing at 340, so it must be something else. And we will just deduct the absorbance at 340 from the absorbance at 260 to get a corrected value, you know, which is more accurate. You know, this is how it works. But here's also, you know, what is a blank? Is no specific blank. The blank could be also, you know, your buffer or the water that you are using. That is a blank. There's no difference.
Okay, I am not seeing any other questions coming up in the chat.
Yeah.
And I think you've covered everything. Oh, wait, one new question just came up. How to use spectrophotometry for counting cultures of bacteria?
Yes. Okay. So maybe in the meantime, when I'm explaining this question, maybe Merlin, can you find the link for our application note on the E. coli on the OD600 and place it in the chat? So yeah, that's a good question. You know, typically we do a measurement at 600 nanometer. You know, often we call this OD600, and we also have an instrument that only does this. You know, the Photopette OD600, and another Photopette cell also has 600 nanometer. And so this is the classical wavelengths. And, you know, bacteria cells or any other cells, but also particle, they are actually scattering the light. They absorb a little bit light as well, but they scatter the light. And that means the light doesn't go straight. It's diverted into the direction and goes sideways or goes back or something like this. So the scattered light will not arrive at the detector. So this means if you have more bacteria, which are tiny particles, basically, E. coli, you know, 3 micrometer or something, diameter of 0.5 micron, and the length of 2 or 3 micrometer, as more of those bacteria you have, as more of the light will be scattered to some other direction and will not reach the detector. So this means there's a direct relationship between, you know, the absorbance of your sample and the number of E. coli cells, what we call the cell density, cells per milliliter or something like this. And we have actually studied this. We did, for all these application nodes, we have our own lab, we do our own experiments, and we cultured E. coli cells. And then we took those cultured cells and we determined the so-called colony forming units, with microbiology experiments, plating them out on plates and growing the E. coli and counting them one by one, these colonies. So we know exactly how many cells are in our sample. And then we measured the sample with the Photopette system, and we measured the absorbance, and then we created the calibration. And this calibration is one of the inbuilt measurement types. So it's inbuilt in the device. And so at the end, you just take the Photopette 600. You put, if you want to directly measure in your E. coli culture, you put our sterile a tip on it, and then you insert this instrument into your culture, press, and you already have the cell number immediately. So I should also say here, you have seen the instrument is made from stainless steel. So of course, the stainless steel and everything you can sterilize with 70% ethanol. The normal standard procedures you do in the microbiology lab. So it's very, very easy by using the Photopette OD 600, and we have just posted the application note into the chat, and you can refer to this.
Okay. So we've reached 5:15, and I don't see any more questions coming up, but everybody can see in the chat the www.tipbiosystems.com is the link to our website. There's a lot of additional information there. And if you have additional questions that haven't been covered, that maybe you're too shy to ask to the whole group, you can always email us at info@tipbiosystems.com. So Merlin, would you like to close out the session?
Yes, okay. Thank you everybody for joining, taking the time. And I hope you learned a lot. And like John already said, if you have any more questions, don't be shy to email us. Then we will do our best to answer it. Thank you very much.
Okay, thank you everybody. Okay, bye-bye. Bye.
Bye.